Hello friends, welcome to my YouTube channel. In this video, I will discuss about enzyme. So let's start. In this session, I will discuss about the concept of enzyme and the properties of enzyme like catalytic property, specificity, reversibility, and sensitiveness to heat and temperature and specific to pH. Okay, here the concept of enzyme. You know the enzymes, the biological catalyst. They are organic thermolyable catalysts. They are increase the chemical reaction without change. They accelerate the rate of the chemical reaction without being consumed in the reaction. If you see this picture, there is without enzyme the activation energy is too high, and with enzyme the faster reaction, and here the enzyme the activation energy is low. So there will be a question that what is the activation energy? In simple term, the minimum energy required to start a chemical reaction, that's called the activation energy. And there is some of important term you can note down: the substrate, enzyme, substrate complex, product, and active site. You can note down and or you can read out. Substrate binding site in enzyme. So there is a catalytic site. That is catalytic site. Catalytic site means one or more or one or many sites. One or many sites located near to the binding site. One or more site located near to the binding site, and they perform the catalysis. This is called the catalytic site. Next is the active site. Active site means the binding site and catalytic site together. They called active site. Cofactor site, additional site. The cofactor site is the additional site for the binding of a cofactor. So, what is the cofactor? A non-protein chemical compound in an enzyme that is bound to enzyme. That is bound to an enzyme. It's called the cofactor. Again, so additional site for the binding of cofactor. It's called the cofactor site. And in non-protein chemical compound in an enzyme that is bound to enzyme. So next is the allostatic site. Additional site for the allostatic modulator which involved in the regulation of enzyme. It's called the allostatic site. Okay. Next move to major hypothesis. One is lock and key and there is induced speed. Let's read. Enzyme active site is performed in the absence of substrate. Enzyme is the lock and substrate is the key. Induced feed, where the enzyme active site is only forms in the presence of substrate. There is huge difference that lock and key says that the enzyme active site is performed in the absence of substrate. But induced feed says that this model says that the enzyme active site only forms in the presence of substrate. Okay, this picture also describes the induced feed model. APOENZYME and HOLOENZYME. So here APOENZYME means here there is a very simple term. What is the what is APOENZYME and what is prosthetic group? HOLOENZYME and prosthetic is very important term. So what is APOENZYME? APOENZYME means the protein part of an enzyme, which is called the APOENZYME. And the prosthetic group, the non-protein part of enzyme, and it is a cofactor that is firmly bound to the enzyme and cannot be removed without denaturing. It's called the prosthetic group. Some of the prosthetic group like name biotin. Okay, so you know that now you know that apoenzyme and prosthetic group. So, what is holoenzyme? Holoenzyme is apoenzyme plus prosthetic group. That means the fully functional apoenzyme with the required prosthetic group together they called holoenzyme. Together they called holoenzyme. Okay. The properties of enzyme. Catalytic property. First is catalytic property. Enzymes are active in very small quantity with their catalytic power. They act with small amount which is enough to convert a large amount of substrates. After the reaction, they remain unchanged. The turnover number of enzymes ranges. That's you can note down in your notebook. So there will be a question on in your mind that what is the turnover number? What is the turnover number? The turnover number, the number of the substrate, the number of the substrate molecules that converted, the number of the substrate molecules converted by one molecule of enzyme per second when it is active site is saturated, when its active site is saturated with the substrate, this will be called the turnover number. So again I will repeat that the number of the substrate molecules, the number of the substrate molecules converted by one molecule of the enzyme per second when its active site is saturated with the substrate, then it will be called the turnover number. These are the properties of enzyme. First is the catalytic property. Next move the specificity. Specificity, the enzymes, the enzymes are very specific in their action. Particular enzymes act on particular substrate. Enzymes are also specific to a particular type of reaction. In some rare cases, the specificity may not be too strong. Enzymes show different types of the specificity, such as bond specificity, group specificity, and some of substrate specificity, optical specificity, geometrical specificity, and cofactor specificity. Now, the bond specificity. Let's move to the bond specificity. So here, it's also called the relative specificity. It's also called the relative specificity. Here, the enzyme is specific for a bond. The enzyme is specific for a bond, and for an example, peptidase. Peptidase specific for peptide bond, and for another example, lipase is specific for ester bond in lipid. Okay, there is another term which that is group specificity. Group specificity, that is very important. It is also called the structural specificity, and here the enzyme is specific for a group. So group specificity means it's also called the structural specificity, and here the enzyme specific for a group. If I give you an example, then it will be clear to you. First, start with the pepsine. Pepsine hydrolyzes the peptide bonds in which the amino group belongs to the aromatic amino acid, like the phenylalanine, tyrosine, tryptophan. That's like okay. So next, okay, that is substrate specificity. After the group specificity, here the substrate specificity. So it is also called the absolute specificity. Here, enzyme act only on particular substrate. Here, the enzyme act on the particular substrate. And like the example, arginase that acts on only the arginine, and carbonic anhydrase acts on only the carbonic acids. Optical specificity. It is also called the stereospecificity. It's also called the stereospecificity, and this is the highest specificity shown by an enzyme. So here the enzyme is specific not only to the substrate but also to its optical configuration. L-amino acid oxidase acts only on L-amino acids, not on D amino acids. Similarly, the alpha amylase acts on alpha 1,4 glycosidic linkage of starch and glycogen, it's not able to hydrolyze beta 1 for glycoside linkage of cellulose. It's optical specificity. Next move, cofactor specificity and geometric specificity. Cofactor specificity means this is shown that the enzymes are not only specific to the substrate but also specific to its cofactor. And geometric specificity, there are specific the specificity is very less. Here the specificity is very less because of some enzymes will work with a small range of similar substrates having similar structural geometry. For an example, the alcohol dehydrogenase that can oxidize methanol and N-propanol to aldehydes. It's end. Next, reversibility. Most of the enzymes catalyzed reactions are reversible. Its reaction depends on the requirement of the cell. In some cases, there are also separate enzymes for the forward and reverse reaction. And some enzymes catalyzed reaction are not reversible. So you may note down the all information from here. And next is the effects of temperature. Okay, there are three effects of temperature here. Three effects of temperature. Rate of reaction increases gradually with the rise in temperature until reach a maximum at a certain temperature. It's called the optimum temperature. The optimum temperature is 37 to 40 degrees Celsius in humans. An increase of temperature that increase the initial energy of substrate and thus decrease the activation energy. Increase of collision of molecules. Like the more molecules become its bond forming or bond breaking distance. So there is an you can use this graph also. And next is specific to pH. They're also very much important pH also. And before knowing pH, there is another term that's effect of temperature. You may know that the enzyme uh, will be inactive at a very low temperature, and this is the reason for preserving the food and vegetables in the refrigerator. Okay, that is the specific to the pH. Specific to the pH, and each enzyme has an optimum pH at which its activity is a maximal. An example that is optimum pH is of pepsin is 1.52, optimal pH of pancreatic lipase is 7.528, and optimum pH of salivary amylase is 6.8. So enzymes are specific for their pH requirement. And some enzymes are active in acidic pH that is also below 7, and some are active in basic pH that above 7, and actives and some are often in neutral pH. So it will also vary. So and there will see there also a Taq polymer is that used in PCR reaction. The optimum temperature that Taq polymer used 75 degree to 80 degrees Celsius. So in this very high temperature, then this Taq polymer is also used in PCR. Okay, so it was our session. Okay, thanks for watching. If you like this video, please share with your friends. If you want to get such type of video, then please subscribe and hit the bell icon to get notification of my each video. Okay, goodbye.